Thank you so much for the invitation and for being here. I'm super excited to give this talk today. So my name is Peter. I'm from University of Copenhagen. A lot of what I've done is actually inspired by my time in the US, where I worked with Yuri Busaki, where we did experiments, but that also led to a lot of tool development. His lab is an experimental lab, but there was many opportunities for me because I came as an engineer. I'm going to talk more about this. As Carol just said, to understand the brain, we really need to study this in action. So we need to ideally do this in behaving animals under natural conditions. We're not there yet, but we're slowly moving that way. And then the big challenge is, of course, integrating these multi-scale data from single cells to population to the behavior. And this is a part that we're all struggling with or excited about doing. And that's why it's a big opportunity for us. And part of this we can solve through openness, through open data, through open tools. So shared tools, standards are critical. It's interoperable, reproducible, of course, collaborations. And as I said, my path started as an engineer in physics before I moved into system neuroscience. And I didn't think it would shape me that much, but I realized when I look back, it actually had a big impact on my path. So I've been building a couple of tools, but we also do experiments in a lab, and I run an experimental lab. And today I want to show a little bit of those experiments and the tools we built. And I strongly believe that tools can accelerate science. Some scientists say like it's all about discovery science, and I don't think that's the only way to approach science. I think a lot of people here are going to agree on this. Hardware and software are two different things to do, as some of you might know. But I think the integration is key. We need connectors, collaborators, translators when we're building things. So it's important not to just be alone. Yeah. So in my lab, we do large-scale electrophysiology. This is in rodents, so freely moving animals. We do circuit behavioral perturbation, and this is all in behaving animals, so freely moving. And then we try to bridge this with the analysis and theory, of course, because we want to understand the brain on a circuit level and not on single cells. We also do a little bit imaging. And my daily life is strongly shaped by open source tools, open data, or open standards. We use OpenEFIS, NeuroPixels, Zebra, Kilosort. Use MATLAB a lot, but Python, Heroku, all these kind of tools are a big part of our daily life. So of course, when I look back, it's had a huge impact on me, an experimentalist, that things are open because this is where we can really accelerate and build on each other's progress. So today I'll talk about a few points. First of all, a little bit on some experimental insights. I want to show you how neural sequences are contributing to theta rhythm generation. Then I'll tell about 3D printed microdrives we built on head cap systems for EFIS in freely moving rodents. I'll talk about a framework for characterizing single cells called Cell Explorer, and then Brainstem. Brainstem and the first are preliminary or ongoing work, while the two others are published. Brainstem is an electronic lab notebook for fair experimental neuroscience, and I think this is really fitting for this symposium. It's about making data accessible, easy to share, and collaborative. So going back to the scientific direction here, we ask how neuronal sequences support cognitive functions. And sequences are kind of a newer thing that's being looked at, mostly focused about firing rates of single cells. But there's more and more evidence showing that the temporal paths really matters. We do that for cognitive functions. So we do this studying navigation, memory, and planning, which is strongly relying on these sequences in the hippocampus. We believe these are being encoded. So when we are representing space or information that's encoded in sequences in the hippocampus. And we believe theta oscillation plays a vital role here to actually create this kind of syntax for it. So it allows for sequence to be generated in a structured manner across brain regions. And I think this video here captures this quite well. Here we have a rat running on a maze. This is a silicon probe recording from hippocampus. And I'm showing you 3 of these place cells projected on this path. So this is we call a memory-guided task, but it's just as much a procedure task. And I'm showing you with the activity of 3 of these place cells. And this is not new to anybody here, I think, but you can really see they're very spatially localized, and they're very repetitive, and they're not just repetitive, they also activate in these kind of groups. And this is the underlying theta oscillation that actually modulates cells. So it's not just something I'm making up. It's a very strong modulation. Yeah, and this is kind of one of the way we can bridge single cells to behavior. So theta oscillations are this oscillation in the hippocampus that's variable depending on behavior. We see it during behavior and during REM sleep, and it's a 4 to 10 hertz oscillation. It's generated in the medioceptal, at least that's what most evidence suggests, and projected to hippocampus. And then in the hippocampus, we know to some extent there's some kind of phase distribution across the septotemporal axis, but just as much across C1, C3 axis. So it's not just a standing wave that's moving together. It's actually highly dynamic. And it's been described as being a traveling wave. So if we go back to the medioceptal, we can see it's been generated by, we know 3 main cell types are participating. We don't know so much about how they actually are involved. Most people believe the good GABAergic population are most important because this is the fastest population. And they're also the one we believe projects the theta oscillation to the hippocampus through disinhibition. So it's actually results in excitation. So one of the question we ask here is if we have this kind of topographic organization in the medioceptal, like Carl Svoboda mentioned for the locus coeruleus, actually. This has become more and more evident across the brain that it's actually highly spatially structured. So we are pursuing this with neuropixel recordings. Here I'm showing you our neuropixel 1. But what's really nice about this is that the coverage of the electrode really fits across the whole region of the medioceptal. So we can do this now with not just 1 shank, but actually 4 shanks in the medioceptal and also record in the hippocampus so we can compare the LFP output in the hippocampus or place cells to the medioceptal activity. And here I'm showing one of these recordings where each circle is a neuron projected on the span of the electrode. This is the average wave from one of these. Recorded with the probe, this is a positive spike, but you can see we can capture it across many sites, so this is important for the extraction of the neural identity spike sorting. We can also see this is a cell that's rhythmic, so it's oscillating to theta, and we can see it has a preferred phase across the behavior. This is like an average. Now, we look across the population. This is neurons recorded at the same time, so 150 cells in the medioceptal. What we can see is that there's structure to it, and we can see a lot of cells oscillating, but it doesn't really look like a spatial structure at first. But if we sort them by their mean phase, so in this case we didn't have hippocampal LFP, so we sorted them by their phase relative to the population rate in the medioceptal, we actually see these very, very nice waves. And again, this is not spatially, this is just across the population. But we can see these waves are also represented during REM at a much slower pace, and now 4 hertz. But actually during non-REM, they seem to fall apart. There we get irregular or more like synchronous delta activity, which might be because it's driven by cortex or something. We don't know at this point. But these are like the 3 states of the medioceptal, or actually only 2 states. Yeah, and then we can look across this population. Do neurons have a preferred phase? What is the phase distribution? And we can see, I'm going to skip first, but I'm going to go to the top. Basically, there's a very wide phase distribution across the cells. Each dot is a cell, and then I highlighted 4 here below. 4 of these rhythmic cells that are strongly theta oscillating, just to show that there are a wide range of phases. And we can actually look across behavior, so a continued trading on the same maze, and there we can actually see that these traveling waves becomes more and more significant across time. This is just for one animal, keep that in mind. But basically, the spatial topographic organization becomes clearer with exposure or trained or potentially just improvement on the maze. This is, we don't know exactly what's governing this. But at least you can see that's a very nice waves now with a continuous exposure across the same population. So this is all so far single metrics. We want to take this to like population or ideally circuit level. And the first thing we look for is PCA, principal component analysis. So what we do is we take the spikes, we convolute them with a kernel, we C-score them, and then we compute principal component on this, and then plot the first 2 principal components. And because we have very wide phase distribution, what it actually comes out to be is a circle. So this again shows us we have a population response. The activity is moving through the population, because if they were all synchronized, which is so far the governing theory about theta generation, they would all be lying on a line and just oscillate up and down together. What we get is actually this very nice rotation. And if you look at a single trial, it looks something like this on the right. This is one behavior, so this is individual theta cycles. So we can really see it's a rotation across the population, or at least sweeps, or there's structure to a temporal structure. And we can see this temporal structure is maintained during sleep, so during REM. We can see it also has a circular structure. It's not as clear, but actually here the same underlying structure is maintained. But then during non-REM, the activity looks very different. It looks more like a single point. We can go back to those 4 cells I showed you before that had 4 preferred phase tuning. And now we can plot the density in this PSA space, because this is a nice thing about PSA. It's the bad thing as well, it's linear. But the good thing is you can actually project your data into it. So here project the activity pattern of 4 of these cells, and you can really see they have 4 distinct places they prefer to spike in the oscillation. And we can also normalize them by the rate. And then we can kind of see that of course this is purely descriptive, but we can see they have a preferred phase in this PSA space, or this kind of population activity. So one thing I didn't talk so much about is what medioceptal theta actually encodes. And we know in the hippocampus that it's modulated, or what is driving it. We know it's modulated by speed, for example. So both the frequency goes up. This is a hippocampal recording. The frequency goes up as animal runs faster, but also the power. So you want to see how does that look like in the medioceptal? Now, actually, it's not that clear. This is a single, trial. And the first 2 components, which is actually what captures the oscillation, there's very little correlation to speed. So it's not like when the speed goes up, the animal runs faster, that the circle becomes bigger. It's actually what turns out to be in a third dimension. The third principal component actually captures much more about behavior and speed of the animal. So the animal runs faster. We still have oscillation, but they change in this kind of PCA space. We can also see acceleration matters, so it's really not just speed, it's more things. And what I didn't show you, that medioceptal also encodes arousal level, many other factors that we need to take into account. But this is like an initial part. And then I'll go back here. What actually is interesting is the third component, the first 2 components seems to capture the rotation. We haven't quantified that well yet, but this is what we can see. So it's kind of like sine and cosine for an oscillation. While the third and the fourth is actually one capturing the speed, acceleration, all these other behavioral changes in the data. And if we go back and look at the single cells, this kind of potentially makes sense, because what we actually see is a lot of the cells are positive correlated with the changes in running speed, but we also have a lot of population going down in rate as a function of behavior, running speed. So the whole circuit is changing the balance across the population. Yeah, so this is some of the experimental data we're working on in the lab, and now I want to go on to some of the tools we built or are building. And the first one is this microdrive, and microdrives are a way to adjust. Once you have put these silicon probes into the brain, I don't know, I hope everybody is familiar with those. We typically want to be able to adjust them inside the brain to record from a different population. And these were first came around for tetrodes and single wires, but this later developed for silicon probes. But now silicon probes are very expensive recording devices. They can cost up to $1,500 a piece. So you typically want to reuse these. So we built, microdrives also gone through these kind of jumps in development, because around the 2010s, the 3D printer came along, and this is where we could introduce 3D printed microdrives in plastic. But then actually what came around later was we can also do 3D printing in metal, not in the lab, but you can send it to a company. So we came up with this idea to make recoverable and reusable microdrives for silicon probes to basically be able to drive down the cost and have reliable insertion and stable recordings. And they look like this. They're very, very simple. But the main thing we introduced was this removable base. So when we implant the probe, we can also extract it afterwards very easily. We have this nice illustration for it. And this is a collaboration I did with Michi Voroshlakosh from my previous lab. So it's a very simple idea, but actually it works really well. And the other thing nice about metal, I mean, it's more heavy, but you can bring down the volume. But it's also very stiff material. It's up to 30 to 100 times more stiff than plastic. So it's ideally it should be a more stable way to record. We also built 3D printed mouse caps for mice. They're basically a physical barrier for the animal, for the probes or whatever you put in the animal, with a Faraday cage to give electrical shielding. And the main thing here, again, make something that can be used across methods. So it's really just the open space that's protected. And we did the same for for rats. And here we need something more structural solid. So this is a much stronger construction, but built around the same principle. It has this kind of folding mechanism, so you can actually go back and open up your head cap after you implant to be able to make adjustments if is needed. And to say before that, we would build these, from scratch with copper mesh and dental cement. So this is a way to speed up the recovery and the surgical procedure themselves and many things. And we made this available for open access, both the files and made a documentation for how to build it and made it available to researchers. And what happened is that when we put it on bioRxiv, a small company in Germany actually reached out and asked us if they could make this or sell it. So we were a little hesitant, but actually it's been really good. So now they're actually selling all our devices, both the microdrives and the 3D printed headgears, and you can buy it for a really cheap price, so you don't have to spend time on it yourself. And I think, again, this actually accelerated it and made it much more accessible for other researchers. And they told me last week that it's now been used in 95 labs, and I don't think that would have happened if it wasn't through a third party. And they actually built and developed on top of this, so this, I'm not sure it's going to stay fully open source, but it's still nice to see. So now they made this is more like a shuttle drive. It doesn't have it's a rail. They called it R2 rail. It's a kind of same idea with the microdrive, but now for NeuroPixel, where you don't need to move the probe after insertion, but you still want to be able to recover it. So now they made a design that's really made for NeuroPixels, and they also improved the holder for its access. So it's nice to see other people are building on your work. Yeah. Okay, so this third part I want to tell about is Cell Explorer. And again, this came about when we were doing experiments and we were analyzing our data, and we realized there was no standard pipelines after the spike sorting. So there was nothing. Everybody had their own way of doing things. Everybody applied filters differently. It was a jungle. And realized that there's ways to actually make this more standardized and faster and efficient across people. So we built this framework, and the idea was really to take the diversity of neurons from the electrical features and project it and correlate that to electrophysiological features. So these are things like simple things like the location, the autocorrelogram, which is really nicely captures properties of single cells. And then already here we can do putative separation of interneuron in blue and in pyramidal cells in red, just purely on the waveform width. So it's really, really simple. But we can also look at other things, like what we call secondary level, like monosynaptic connection, how they respond to some kind of external device, oscillations, and state transition in the brain. So all of this can both characterize cells, but also help us to do separation of the cell types just based on electrophysiological measures. And the third level is, of course, behavioral measures and states. And very briefly, what it's about, I'm going to show you 2 videos. But basically, it's a pipeline that takes all these kind of metrics. It takes the raw data, metadata, spike sorted data, and then it puts it in a standardized structure, where the 2 most important ones are the cell metrics and then the metadata going in, and also the output. And then it's really about this graphical interface to make it easier for people to explore these metrics. And it's all built in MATLAB, so it's not fully open source. It's built on proprietary software. But everything we built is on GitHub. And there's a dedicated website. I'll show you in a minute what we have there. And it's really, besides the format, it's really 2 graphical interfaces. This is one we call Cell Explorer. This is an interface for looking at these single cell metrics. So right away, people run these pipelines, and they can see like this, the waveform characteristic, the firing rate characteristic, at time and across the autocorrelogram, for example. We can look across the population up here. So now this is grouped across maybe 20 or 60 sessions of data. But it gives a lot of easy access to your data. So right away, you get all these kind of plots, like the waveform, ACG, ISI, firing rate, how this responds to triggered average. You give that input in, connectivity matrix from monosynaptic connections, and it has a lot of built-in plotting methods. And I'll show you a short video just to show it in action. So basically, it's an interactive GUI where you can click on any kind of points or line, go to that, you can highlight, you can show measures from these cells. And people so far have been really happy about it. This is monosynaptic connections. I'll just show one more and then I move on. This is the connectivity graph examples. These are modules that we found in the hippocampus. So these are actually cells projected through monosynaptic connections that you can very easily explore, select, and so on. I'll move on. And the other GUI is Neuroscope 2, and this is named after the original Neuroscope, which was 10 or 20 years old. And the main thing here was to take all these kind of metrics now we had, or standard containers. I didn't say so much about it, like behavior, but cell metrics, all the things, and be able to go back to the raw data and actually explore the raw data, but also directly work with the raw data, because there's not that many tools besides that are easy doing this. So this is hippocampus recording. Basically, it's a really, really nice interface for expecting data, both pre-spike sorting and after spike sorting, looking at behavior, looking at like physiological events in the brain. And again, there's a small video that I'm just going to play you. Again, all in MATLAB, but it's actually extremely fast. Extremely fast. It is very fast. It's much faster than original software. So you can do online spike detection. So you can do this across your channels. You can do. You have the electrode groups. This is if you have multi-shank electrodes. You can do tags, so you can highlight channels very easily, share that cross-analysis. You can quickly do nodes. You have access to the epochs of the data. So let's say you do some kind of behavior and sleep or something. All of this is accessible right away. And then you can also work with the spike sorted data that you created. And now you can also extract waveforms from the trace and go back. Much better, have a much richer description of your underlying data as an experimentalist. You can population rates, many things. Also spectrograms, noise across channels, RMS across channels, and many features built in. And we finally built this website hosted on GitHub with links to the code itself and the paper. And then has a bunch of tutorials to get people started, has documentation of the data structure, the processing module, graphical interfaces, and then of course also some of the data we actually shared at that time from my previous lab. Yeah. Okay, and the last thing, I can't see the timer. The last part I want to talk about is Brainstem, and this is something we're actually developing. As mentioned before, this is an electronic lab notebook for experimental neuroscience, but it's really about bridging the people who's collecting the data to everybody else later on. So it's about getting experimenters to put their data in and then share it afterwards. It's really about the whole thing. And it's a centralized solution, and we built this as part of a U19, oxytocin U19 in the data core there, where Alyssa and I are part of it. I'm co-director of it at NYU. And then there's been other couple of people involved, but actually also a company solution. And this is, I think, rare that you actually have money for it. But this was nice about this U19 that we could actually support like professional development of it. So we can both build this, but we also had professional help both with web design and some of the like technical implementations. So I said this before. It's really a web-based notebook solution. It's centralized, meaning we host it with a service called Heroku. It's built on Django, which is in Python, so it's research-oriented. And it's really about like a notebook for explaining neuroscience data. We're focusing on neurophysiology, and it's tailored to that. So what that means is also that it's a structured format, so it's not like they have a complete open slate. When they describe the animal, for example, they have a list of strains to select from species. Everything is structured. If it's like which brain region you're putting your probe in, this is also true, like for example, Alan is to atlases. So it's everything is linked, and it's the idea is to create a rich representation of the data. And this is really because without the metadata, and again, we're not hosting the data, it's only hosting the notebook aspect, which is really metadata. You need that to understand someone else's data. Without that, it's impossible to read someone else's data and make sense of it. And we need that already from the experimenters when they're collecting the data. So this is a tool for them, but it's really also a tool for the community to accelerate neuroscience. And one of the aspects we struggle with is that a lot of times these kind of metadata lives in various sources when people are collecting the data. It could be in lab notebooks, something in spreadsheets, something is with the data, and something is hidden in scripts. So when you get someone else's data, it's actually very hard to know all these things. You typically have to talk with them, and that is a big hinder for reuse. And then the other problem is that often these things are actually personal language. There's a lot of lab convention on implied experimental conditions. So basically, in the end, you have too little information to work with the data, understand the conditions, how it was collected. And this is where the Institute has been the forerunner of this. But this is hard to get that kind of scale in a smaller lab. And this is also what's the focus here, is getting all the data from the smaller labs to be accessible. Yeah, and then, of course, also when you're sharing data, it's really time-consuming if you do it afterwards. So we want to make that barrier as small as possible, but we also want to make it discoverable. And this is kind of a limitation now with current archives. Is you can find the data, but it's actually very hard to discover it. It's not like you're going to stumble on it, or you typically have to know exactly what you're looking for. So I think if we have a rich model accessible online, this can help solve that. And we, again, to get people to use it, we focus a lot on making this user-friendly, standardized, so other people can work with the data afterwards. We want to make it customizable, so it doesn't feel like when you're working in Brainstem that it doesn't fit your need. So if you're an experimenter, it should feel like this is fitting your experiments. But of course, also science is not done within labs. So we want to make it collaborative. So we're building a sharing mechanism where you can share that with other people in the lab, outside lab, with read and write permission. So it has this kind of granular permission level. And then it has the API as well, so we can have it machine readable and not just human readable. And this, I think, is really powerful as well. And then the underlying model itself is relational. So that means we are, instead of giving people these open notebooks, we're actually giving a relational model. So they have to understand the model to actually put the data in. But this also allows us to work in a modular way. So when you're putting things in, I'll show you in a minute, there's a structure to it and a place where things go. And that's ideally to make it flexible across different types of experiments or data collected or animals and so forth. And then you can also take it a step back and say why we're building this in a greater scope. It's really to address this overwhelming volume of data that is never being reused. We want to put it in there or give option for people to put in there. We want to streamline the discovery and accelerate science. We want to lower these barriers for collaboration. We want to enhance reproducibility and transparency. And then, of course, also align with the funding agencies' priorities, like the NIH and EU sharing initiative. And then, of course, we want to make the data fair. So findable, accessible, reusable, and interoperable. And now I'm going to show you the model. And I know this seems like a lot at first, but this is actually is not a very complicated model for what we're trying to build. So on the top, we have projects. Projects is a way for grouping the data. This is where permission exists as well. And already here, we have subjects and sessions. Sessions are the data collection or the actual experiments you do. And sessions are subjects, of course, your subjects. We have groups for these, either cohorts or collections. And then below that, this is color-coded by the same things. So when you're working with a subject, you can do procedures on the subject. You can have logs, which is like simpler things. And the session is built up of the data acquisition, any manipulation doing, something that's special from just natural states, and then the ideal behavior of the animal. So in each of these boxes actually, has tailored data tailored model that fits tailored metadata description of the different types of data. And this is also what makes it scalable or flexible, is that you can describe extracellular data with one type of metadata, intercellular video with another type, and the same for procedures. We can tailor this kind of metadata around procedures. At the same time, we can offer a backend with reusable resources. So we have consumables, hardware devices, suppliers, for example, and we have a taxonomy, so like strains, species, and atlases built in, so the users also get something right away. And I won't spend so much time here, but this is the permission model. And the main thing is that permissions are primarily set on project level. Everything else inherits. So it's really meant to be just one place where you go in and share it with other people. And you can share on read and write. And then within, you can have members, contributors, managers, and owners. And then we built a support and documentation website where you can also submit tickets. You can tell if there's something not working or if there's something new you want to have. Yeah, and then I think we have time for the demo. So I'm going to jump in. And if you go to brainstem.org, you can actually do the same thing. I need to go out of the presentation. So if you go to brainstem.org, I'm running this locally, but it looks the same way. This is the landing page. So this is what the landing page looks like. And once you create a user, you can log in to your dashboard. And this gives you an overview of the data you have access to. So here you can see, I can see my projects, I can see subjects, sessions, and I can also see other things like data storages, inventories, my experimental setups, and statistic about shared things like strains, the most popular strains, hardware devices, and so forth. But let's go create a subject. So everything is through forms. And I already created a project for today. So now I'm creating a subject called Odin Subject 1. I'm going to tie to the Odin project, and that basically means people who access that project can now also access the subject. And then here you can see some of the standardized fields. So here we can see I want to put in as red, for example. And I can tie to, you can say it's a long event. So everything is like standardized. So these are actually relationships that you're creating. And this is now we have a subject, and you have a simple description field. You have the field I mentioned above. You have the genetic line, the birth date. All of these are standard fields. But also, like I want to show you, we have procedures, for example. So the subject, when you do stuff on your subjects, this is what we call a procedure. And we can pick here from available procedures. So let's do a silicon probe, for example. You can say when it was implanted. But most important, you can also pick from available probes you have in your inventory. So we have a standard list of probes, but then you create a local, inventory list of your probes or your viruses or your optic fibers. So basically, you're working with these internal lists afterwards. We can also tie it to a setup. So let's say I want to build let's say I implanted this probe with some kind of equipment. I don't have anything relevant here, but I can tie it to a setup where things are happening. And then I can link to the atlas, and this is, again, using external sources. So this is my preferred atlas, the Allen Mouse Brain Atlas. And I'll have atlas regions from that. But I can actually change this very easily. I can pick one. Let's say I want to have a red atlas instead. Now I have the brain regions from that atlas instead. And the same thing about the consumables up here. If let's say I do an optic fiber implant instead, I won't get a list of silicon probes. I'm going to get a list of fibers instead. So things are adjustable or like flexible to users. Same thing about coordinates. If I have a preferred way to put in coordinates, I can actually change this. So let's say I put it in. Let's say I put it in hippocampus. So I can say save. So now I created a subject. I go back. I created the subject with this procedure, and now I can also tie it to a data collection session. So this is typically where you would collect data. So again, a data set is made up by the behavior, data acquisition, manipulation epochs. Epochs are a temporal aspect of your data. But basically, you go to, let's say you want to say you collect a data session. I'm going to do extracellular recordings. I'm going to do the recording on this setup. And now I can also pick that probe I just put in the subject. And this shows the power that I'm actually creating relationships all the time in the database when I'm making these selections. So now I created a data acquisition. You can see here these fields are customized to extracellular recordings. And you don't have to put all this in at first because we have the APIs. You can actually sit in Python or MATLAB afterwards and fill out the rest, so to say, from the data if you create scripts. Yeah, this is kind of what the main overview I would give you here. And this is accessible now. It's not really fully production yet, so we appreciate any kind of input. So finally, this is the people who's been involved in my previous lab with Neurophysiology. He played a big role here, of course. With Mishi Voroslokosh, I did the 3D printing together. Ming Xie from my current lab did a lot of the documentation and part of the project I showed, the experimental project. And these are the current lab members of the lab. Thank you.
Thank you, Peter. So we'll have like a few minutes for questions. And people who have questions, someone will pass the microphone to you. So please raise your hand.
Okay, I had some questions, but to go back to the hippocampus work, from a scientific question is, you showed that the frequency goes up as the animal starts to run, and how does that manifest itself as shifts in timing along the hippocampus? Is it changes in lag time, or is it changes in phase of the oscillations?
We actually didn't do the spatial recordings yet, so I know Jack Tish from the Busaka lab has done it, and there's another paper, a science paper that did look at this. I don't think they looked at speed, how it changes with behavior, so I wouldn't know.
Okay, and then
but we can do that in the medioceptor right now, and potentially we have the force shank, so we don't have very good spatial cover at the hippocampus at this point, but this is stuff we could look into.
Then I just had a technical question. I mean, you did your principal component analysis in space or electrode position and time. I was wondering if you considered doing it in space and frequency, because you showed rotating vectors, and that would just turn into a single component in frequency space, and the phase would give you the rotation.
Yeah, this is a good point. We haven't done that yet. We also used the zebra. I didn't think I had time to show it, but we used zebra as well, one of a nonlinear wave to project the data into lower dimensional space. And there, what we found was actually that you can decode the position of the animal. Not as good as hippocampus, but if you have this task when the animal's running left and right, and you're feeding these labels, if the animal's on the left or right side, you can actually train zebra to distinguish where the animal is. So we're looking into how that's done right now. This is, again, very preliminary.
Was that a joint embedding of the behavior and just like using electrophysiology, completely separate?
Yeah, you train on state labels, left or right side, and then you give the spike data. Okay. Also convoluted, yeah.
I think I may be next. Related to David's question, could you kindly tell us where the place cell or the location information was decodable in that interesting three-dimensional PCA representation? In other words, was it in the first two components in terms of some phase, or was there extra information in the third?
Again, this is very preliminary. We did look across the shanks to see one caveat here, and again, this is because of preliminary. The lateral septum above is known to have spatial information. So you have the medioceptal, but you actually have lateral septum above, and there we know the cells are encoding spatial tuning. But looking at the data, we looked across the electro, and there was no cells, there was no spatial structure to which cells had information about it. Yeah.
Further questions?
Is there any spatial organization of anything you've seen?
We saw the spatial organization I showed you there in the beginning. What we have here, this is actually sorted by the position, not sorted by phase. So this is the spatial information we have at this point. There's also spatial organization in terms of cell types where they are, but this we haven't looked into yet. Like apergic cells are more across the center of the medioceptal. for example. And we're also looking across if this relates to the cholinergic population or which cells are actually encoding, let's say, speed or other measures.
But this does not appear genetically encoded, right? This does not appear in session 1. This seems to be a learned topographical map.
Yes. Yeah, that's true.
What do you think we could do to improve reusability of those tools? What I see in Neurophys, there is even more like this. Oh, this is mine. These are my approaches. It might be as successful as yours to share, but typically they're not. So they're, instead of even reusing somebody else's data or somebody else's tool, they would still keep developing their own things, which are maybe suboptimal. So how could we inspire more reusability? And even in your case, let's say, did you look for data sets which could provide you support for whatever you found, right?
Yeah.
How could we improve those archives? You said we are not providing help to discover things, right? So again, how could we make this circle of discoverability in existing resources and reuse even?
Part of this is that I think we need to have richer metadata, so people are more likely to use your data. So we ran a course last week in my university on open neurophysiology, and one of the things people say, and I'm actually surprised, is they say, "Oh, I don't want to waste time using someone else's data if it doesn't work, or it doesn't have what I need." And I think that's a huge mistake, because you're saving so much time by reusing that data. You don't have to spend years collecting the data. I think it's a change of mindset, and I think we need to provide more description of the data. I noticed when I look back on some of the data my previous lab shared is the data without modulation, manipulation, anything. This is the one that's really popular. It's really simple, just running left and right. As soon as you do any kind of specialized things like manipulation or freezing or sphere conditioning, the data has much less, reuse value. At least the wider audience is not going to pick it up. I think we have to train people to know it's there and understand that it's not actually a huge hurdle to jump on someone else's data. And
you reuse data of others?
Yes, not like for Cell Explorer, we actually work with both data from Nick Steinmetz and Josh, and from the Allen Institute. So we actually reused it on data on both of these data sets. And one of the kind of discovery we did was the NeuroPixel has a very different hardware filter than the Intan chips, for example. So we were actually losing a little bit of this information about cell identity from the spike waveform, because it has a very aggressive hardware filter. And this was actually fixed in version 2. I mean, now it looks much better. Now we can actually classify from waveforms from the data. But these are kind of things you don't know if you have using someone else's data, of course. This is why people are hesitant, I think.
Yeah, I mean, I can't agree more about the importance of metadata. And I think there is actually a huge hunger for this data, right? So I think Saskia will talk about this a little bit. And I think for some of the neurophysiology data, there are probably 50 papers published by outsiders for each paper published by the Allen Institute on the neurophysiology data. But I was going to ask a little bit more about reusability of the kind of the apps and stuff like that. Obviously, MATLAB isn't ideal, but I wonder if these sort of desktop applications that do all are really if that's really the way to go. And I mean, for your lab, of course, maybe, but sort of for sharing and also, maintaining this thing, right, over time, right? Everybody will want to use their own.
You're afraid it's not scalable, or it doesn't fit into like environment, like a shared environment?
Well, so first of all, you have to always download the data and figure out the data formats that the choices you. With standardization of the data, this will become easier, right? But then you have to write this, but there are of course so many of these. Everybody has a desktop tool like this, right? So what's the magic there? Why not do a web app? There you'd be
it's a cloud solution.
But that you'd be at least the first in class, and so I'm just curious, are you thinking about that? Same for the metadata capture tool. There's a lot of metadata capture tools, and you've tied it sort of to a metadata. Homebrewed metadata format. Again, how are you going to make this live? How do you make it? How do you get adoption?
generally you get adoption if people are saving time or you introduce something new for them. And this is not really the case with notebook solution. There you typically need, people have to see right away, okay, this is nicer for me. Or you do, I do think you do, but not on the first experiment. You do save time once you have it in some kind of standard. So you need to have someone who has a little bit longer perspective on where you make sense to invest. And then for the cloud, I think that's still a big barrier for a lot of experimenters to put their stuff in the cloud, to work on a terminal only, for example, or like a certain thing. It's hard to make the graphical interfaces as fast if you're working on cloud data, for example. Maybe you can nowadays, but when we were doing this, for example, some IT there was trying to help us set it up, and the way the stuff they could make for us was like 10 or 20 times slower. And that hinders, then you prefer working on your local computer. And I think people are also afraid of giving away that data for free, like not for free, sorry, putting it on the cloud and not really sure about where it goes or what it's going to cost. If you have it on a local computer, you know how much it's going to cost you because you paid the hard drive from upfront. Or if the university pays for storage, that's also a free solution.
I would say it's a hidden cost.
Yeah, but you don't feel it right away.